So our final section of this chapter will be on hemostasis and platelets, which is the control of bleeding. Along with the evolution of a circulatory system, we had to stop potentially fatal leaks. Hemorrhage is the condition of excessive bleeding. We'll begin by looking at platelets themselves and then the three homeostatic mechanisms, which are vascular spasm, platelet plug formation, and blood clotting itself, or coagulation. Platelets are important in all three, so let's begin by looking at them. Platelets are very small fragments of megakaryocyte cells. They're about 2 to 4 micrometers in diameter, and they contain granules. They have a complex internal structure and an open canalicular system. They use amoeboid movement to move around and do phagocytosis. A normal platelet count is about 130,000 to 400,000 platelets per microliter. Platelets have a variety of different functions. First, they can secrete vasoconstrictors that will help reduce blood loss at the site of the injury. They'll stick together to form platelet plugs that seal small breaks in the skin. They'll also secrete procoagulants, or clotting factors, that promote clotting. They initiate the formation of clot-dissolving enzymes. They'll also chemically attract neutrophils and monocytes to the site of inflammation so that they can do their jobs. They'll phagocytose and destroy bacteria. They'll also secrete growth factors that will stimulate mitosis to repair the broken blood vessels. Now, platelets are produced by a process called thrombopoiesis. These stem cells that develop receptors for thrombopoietin become megakaryoplasts. Megakaryoplasts repeatedly replicate DNA without dividing. That forms a gigantic cell called a megakaryocyte with a multi-lobed nucleus. They're about 100 micrometers in diameter, and they remain in the bone marrow. Megakaryocytes live in the bone marrow adjacent to the sinusoids. The long tendrils of cytoplasm, or proplatelets, protrude into the blood sinusoids. The blood flow will split off fragments called platelets. These will circulate freely for about 10 days. 40% of them are going to be stored in the spleen. This figure shows this formation, where we'll have proplatelet tendrils reaching into the sinusoid of the bone marrow and dropping off little platelets. So, vascular spasm is a prompt constriction of a broken vessel. It prevents it from leaking out too much blood. It's the most immediate protection we have against blood loss. Vascular spasm is a result of pain receptors. Some directly innervate blood vessels to constrict. If we have smooth muscle injury, it will cause vascular spasm in the same way. And then platelets also release serotonin, which itself is a vasoconstrictor. The effects are prompt constriction of a broken vessel. The pain receptors have a short duration. In minutes, they're sealing off the vessel. Smooth muscle injury, well, it'll take a little bit longer, but they'll still have the effect. This provides time for clotting to occur. The platelet plug formation is where we see the endothelium. So platelets do not adhere to the endothelium, the inner lining of the undamaged blood vessels, but when the endothelium is damaged, we'll see that it has collagen fibers in its wall. They don't have the protection of this prostacyclin protein. And so when these collagen fibers are exposed to blood, the platelets grow long spiny pseudopods and adhere to the vessel and other platelets. The pseudopods then contact and draw the walls of the vessels together. And the mass of platelets that forms is called a platelet plug, which ideally will reduce and stop minor bleeding. As the platelets aggregate, they undergo degranulation, releasing a variety of different substances. First of all, serotonin, which is a vasoconstrictor, and then adenosine diphosphate, which attracts and degranulates more platelets. thromboxane A is also released. It's an eicosanoid that promotes platelet aggregation, degranulation, and vasoconstriction. And so we have a positive feedback cycle activated until the break in the small vessel is sealed. And then there's no degranulation, less production of thromboxane, so on and so forth. Finally, there's coagulation. In coagulation, we'll see blood clotting. This is the last and most effective defense against bleeding. It's where we're going to convert the plasma protein fibrinogen into an insoluble fibrin thread, or a framework of the clot. The procoagulants, or clotting factors, usually are produced by the liver and are present in the plasma. They activate one factor and it'll activate the next to form a reaction cascade. We'll look at that briefly in a moment. There's an extrinsic pathway where factors are released by the damaged tissue, begin the cascade, and then an intrinsic pathway where factors found in the blood begin the cascade, like platelet degranulation. Here's a scanning electron microscope view of a blood clot. You can see the framework of fibrin and the platelets filling up that framework to form the clot. Here's an example of enzyme amplification that goes on in clotting. There are many different factors involved. There's a figure in your textbook that'll give you much more detail. But as one factor is secreted, it activates many more of the next factor, which go on and activate many more of the next factor, so on and so forth, until we see lots and lots of fibrin produced. So rapid clotting is activated by each cofactor, activating many more molecules in the next step of the sequence. Coagulation is completed after we activate factor X. Now, we're not looking at so much detail of each of these factors, but you can see factor X is one in the cascade here. Then prothrombin activator converts prothrombin to thrombin. And thrombin is going to actually convert the fibrinogen into the fibrin. This positive feedback speeds up the formation of prothrombin activator and thus completing the coagulation. So after a clot is formed, we're going to see that there is clot retraction within about 30 minutes. And clot retraction is where spinous pseudopods from the platelets adhere to strands of fibrin and contract. So it's much like a drawstring closing a purse. We pull the surrounding tissues together, and the clot becomes much more compact. Platelets and endothelial cells will also secrete platelet-derived growth factor, which is a mitotic stimulant. It causes the fibroblasts to divide and smooth muscle to multiply, and that will repair the damaged vessel. Eventually, the tissue repair is complete and the clot needs to be disposed of. This is the process of fibrinolysis. Factor V speeds up the formation of this chemical called kallikrein, which is an enzyme that converts plasminogen to plasmin. Plasmin is a fibrin-dissolving enzyme that breaks up the clot. Now, how do we prevent inappropriate clotting? Why is it that the platelets don't just form clots anywhere? So as we learned earlier, platelets don't adhere to the endothelial layer of the blood vessels because of this prostacyclin coating. So this is platelet repulsion. Now thrombin is another issue because thrombin is circulating in the blood. And as we learned earlier, it's important in conversion of fibrinogen to fibrin. Now if thrombin is just easily circulating around in the blood, how do we stop this? Now in the blood when it's flowing rapidly, this isn't a big issue because it gets diluted fairly quickly. However, if we have slowing of blood flow, then we can see clot formation pretty readily. So when we see shock, such as the heart rate slows, then clots could easily form in the blood. But at normal paces of blood flow, it's not an issue. There are natural anticoagulants in the blood, things like heparin and antithrombin. They interfere with the formation of prothrombin activator, And antithrombin from the liver is going to deactivate thrombin before it can even act on fibrinogen to form fibrin. So there's 3 mechanisms to prevent inappropriate clotting. Here's a great place to shut your books and pull out a piece of paper and see if you can review what we've just covered. Write down as much as you can remember about each of the steps of clot formation. Remember, there were 3 main phases: vascular spasm, platelet plug formation, and coagulation. Now this is going to be difficult. You might not recall a lot, but as you go back and explore in the text and in your notes, you'll be able to fill in the gaps. Keep filling in the gaps until you have the complete story. Now there are several clotting disorders. The first one we'll look at is hemophilia. It's a deficiency in any kind of clotting factor. There are a couple of different forms of hemophilia that involve any of the factors in the cascade. They can shut down the whole coagulation cascade, and thus bleeding can't be stopped. Hemophilia is a family of hereditary diseases that are characterized by deficiencies of one factor or another. There's a sex-linked recessive variety, like hemophilia A that we saw in the Russian and European royal families. There's also hemophilia B, in which we're missing a different factor. Now, you don't have to be responsible for the names of all these various factors, but it's interesting to note how they play their role in the cascade. Hemophilia C is an autosomal case, meaning it's not on the sex chromosomes, that involves a different factor still. If physical exertion causes bleeding and then excruciating pain, transfusion of plasma or purified clotting factors can be used to treat hemophilia. And factor V is now being produced by transgenic bacteria, so genetic engineering. Hematomas are masses of clotted blood in tissues. They're bruises, basically. They're also a family of coagulation disorders, like thrombosis, which is abnormal clotting in an unbroken vessel. A thrombus is a clot, and they most likely occur in leg veins of inactive people. Again, if the blood flow slows down enough, remember thrombin isn't dilute enough, and thus it can initiate clotting. Also pulmonary embolism. The clot could break free and travel from the veins to the lungs and cause an embolism there. An embolus is anything that travels in the blood and can block vessels. We also would see infarction or tissue death occurring if a clot blocks the blood supply to that organ. This is how we see myocardial infarction, heart attacks, or strokes happening. 650,000 Americans die annually of thromboembolism, or traveling blood clots. How can we reduce the risk of this, do you think? Perhaps exercise increasing the circulatory flow would be a good solution. So this brings us to the close of our chapter on the blood portion of the circulatory system. Work with your diagram and see if you can put the whole picture together before we meet next in class. There we can address any questions and help you out with some of the diagramming challenges.